Thank you very much for inviting me. So yes, I want to talk about coordination of cilia today. So the approach that I'm taking is thinking about an analytical framework to connect the properties of an individual cilium to the emergent wave properties when they coordinate into metachronal waves. So that's the angle that I'm looking at this problem from. So just first of all, the introduction to cilia. So there's these active organelles that beat to create fluid flow. So you find them on the surface of microorganisms, and they can be used for propulsion, and you find them on epithelial, for example, in your lungs. But the motors that drive them to move through their beat cycle, they're within the individual filaments. So the question is, if the motors that make this thing move are within the filaments, how do the neighbors know how to talk to each other? So the motors are very much separated from each other because they're within those filaments. And just about thinking about coordination, cilia are in a lot of different places in the human body. So they play a role in symmetry breaking in embryonic development. They move signaling molecules around the brain. They transport over through the fallopian tubes. They move mucus out of the lungs. And as I said, they propel microorganisms. So we want to understand how cilia coordinate, so we know how they can work properly, because as you can imagine, when they don't work properly, it can cause all sorts of problems. So the type of coordination we see when we're thinking about systems with many cilia involved is this metachronal coordination. When you have systems where you maybe just get 2 or 3 or a small number of cilia, you see different types of coordination patterns. So you might see synchronous beating, you might see other types of patterns. And just to highlight, Kirsty Wan's group is she does experiments looking at lots of different ciliated species. So if you want to look at small numbers of cilia and how they all do their thing, you can look at some of her results. But what I'm thinking about here is metachronal coordination when you've got lots of cilia. So what metachronal coordination means is, if you imagine the beat of an individual cilium, it moves through its beat cycle, and we can characterize the position within that beat cycle as a phase. And as you move along the tissue, you can look at the phase of the cilia as you move along. And the metachronal wave is a wave of the cilia phase. So we're not talking about a wave of fluid flow. So the cilia create fluid flow, but we're not thinking about any kind of wave in the fluid. We're thinking about a wave of the cilium phase. So this is illustrated here. These arrows on the screen, they show the position where the peak of where the tip of the cilium is at its peak. So you can see a peak at one arrow, you can see a peak at the next arrow, and that characterizes some wavelength. And you can see as you move through in time that the peak position moves along. So we've got a wave. So that's what we're looking at. And as I said before, I'm trying to develop an analytical framework to connect the properties of the individual to this emergent wave. As this slide has just said. So we're thinking about the physical properties of the individual cilium, so the geometric properties, and then how that links to the emergent wave. And the model approach that I take is to model each cilium as a bead moving around a circular trajectory. This is called a rotor model. It's commonly used in these types of cilia problems. And there's some experimental results from about 2010 from Ray Goldstein's lab that showed that for a Chlamydomonas, you can use this rotor model quite nicely, and you can reproduce the fluid flow very nicely with this rotor type model. So it's a commonly used approach, and it does a nice job in recreating flow fields. But the reason why it's nice from a theoretical point of view is because we can calculate the hydrodynamic interactions between the cilia. So we're thinking about hydrodynamic interactions as the mechanism for coordination between the cilia. There are other methods that cilia can coordinate through, but hydrodynamics are ubiquitous to all systems, all ciliated systems. So that is the method that I'm focusing on here today. So we can, if we model each cilium as this bead, this sphere moving around on a circle, we can calculate the hydrodynamic interactions between them using Stokes flow solutions. before I go more into the model for the many cilia problem, I just want to mention hydrodynamic synchronization between two cilia with this rotor model. So this is a well-studied problem. And so you can ask, well, what is needed to achieve hydrodynamic coordination if you've just got two cilia? And the key thing when you've got two is you need to break the permutation symmetry between the two cilia in order to get some kind of coordination. So what that means is that there has to be some kind of physical difference between the two cilia. So that could be if they're experiencing a different drag coefficient due to the surface, so a different amount of friction on them. If there's some flexibility of the anchoring, so they're not rigidly constrained to the trajectory, or if you have a non-uniform beat pattern. They're all examples of how you can break the permutation symmetry. But the main thing is that there has to be something physical that allows you to tell the difference between these two things. Just moving on to some simulations. So these were done by this figure you can hear was done by Doug Bromley. So he did a simulation looking at hydrodynamic coordination between cilia, and the waves that you can see here, they show the phase difference. And if we're looking at metachronal waves, what we're looking at is we're looking at a fixed phase difference between neighbors. So when you get that fixed phase difference between neighbors, then you know that you've got a metachronal wave. So the simulations tell us that hydrodynamic coordinations are something that can facilitate this metachronal coordination. But the point of the analytical work here is to understand how. So we know that hydrodynamics can do it, but we want to know a bit more about the details of it through the analytics. So, okay, as I said, we're using a rotor model. So the setup is we've got this bead moving around some circular trajectory. And the main parameter that I want you to keep in mind is the phase angle phi. So phi is telling you how far around this circle you are. So if you're thinking about a filament, it means how far through that beat cycle you are. So we're using phase phi to tell you where this filament is in the beat cycle. So we're looking at how that evolves. Theta and chi, they're parameters in the model that just tell you how we set up the rotor, so the circle that it goes on, how it's tilted, how it's oriented relative to the lattice that we put these cilia on. So we put these trajectories onto a 2D lattice. It's a 3D problem, so the fluid dynamics is all done in 3D above a surface, so it's a 3D half space, but the rotors are arranged on a 2D lattice onto the surface. So we can write down a dynamical equation for each of these cilia. So this is a dynamical equation for an individual cilia. So we're not thinking about the metachronal wave yet. We're just thinking about an individual at this point. So each rotor phi, and we're giving it a subscript I to label an individual rotor. We can look at how it evolves in time. And each rotor has an intrinsic velocity, which is given by the force that drives the bead around the trajectory. So that represents what the motors are doing within the filament. And then this zeta, this function in the denominator here, that is the frictional force. So we make this phase dependent as well, because if you imagine you've got a surface, if you're moving around your rotor, if you're closer to the surface, you might feel a higher drag than if you're further away from the surface. So we include this phase dependence here, and A is the size of this trajectory, the radius of the circle that this bead is moving on. So that somehow represents the amplitude of the cilia beat. Then this second term here that we've got, this encodes the hydrodynamic interactions between all pairs of rotors. Hydrodynamic interactions are long range, so the sum is summing over all pairs of rotor, and we're looking at an infinite lattice, so it's an infinite sum over all pairs. So we've got the force acting on another cilium at some point. So this creates a flow. So we've got a force that's driving around a different cilium, and it's going in this direction t, which is the tangent to the trajectory. So that just tells us the direction that this force is going in. This tangent here is projecting the force from this other cilium onto our cilium of interest. And then for the Green's function here, we use something called the Blake tensor. So if you have bulk fluid, if you don't have a surface for your Green's tensor, for your Green's function, you could use the Oseen tensor, and that encodes the hydrodynamic interactions between two pairs. If you've got a point force here, what flow do you feel over here? But then if you put a surface in, you have to ensure that you've got a no-slip condition on the surface. And if you want your no-slip condition on the surface, then you use the Blake tensor. That uses an image system to ensure that. So we use the Blake tensor here, and that gives us the hydrodynamic interactions of all pairs. The force and the friction here, these are periodic. So we write these as a Fourier series, and we leave these coefficients in the Fourier series as parameters in the model. And they're going to show up, and we're going to think about which harmonics show up later. So when you see ANs and BNs, CNs and DNs later, that's where they're coming from. So that's the dynamical equation for an individual cilium. But then we've got to ask, how are we going to connect this to the emergent wave? I'm just going to skip over this very quickly, but just to say that it's reasonable enough to use CN and DNs as 0, so we can have roughly a constant friction in using the approximations we're using here. And those approximations are that we're assuming that the cilia are sufficiently spaced apart, so the gap between cilia is large compared to the size of the rotors, and the gap between cilia is comparable to the height above the surface as well. So those are just two model approximations that we use. So the next few slides that I want to go through now, these are the technical steps that allow us to go from this dynamical equation that tells us about the motion of a single cilium to an emergent wave. So we want to say, well, how can we get here from this equation to something that tells us about the emergent wave? And what we're going to look for is we're going to look for a dispersion relation for a metachronal wave, and we're going to look at a linear stability analysis. So in these next few steps, if you get a bit lost, just keep in mind that that's what I'm trying to do. I'm starting from the dynamical equation for an individual, and I'm looking for a dispersion relation, and I'm looking to do a linear stability analysis. So I'll talk through those steps, but just keep that in mind. One of the things about a metachronal wave that we're looking for is we're looking for a fixed phase difference between neighbors. That's made slightly complicated by the fact that we've got an intrinsic velocity that's phase dependent. So this is not constant. So what we can do to handle that better is choose a new coordinate, and we're going to call it phi bar, where the speed is constant, the intrinsic phase velocity is constant. So we can write down a relation between the old coordinate phi, which is just an angle as you move around a circle, to phi bar, which is just some choice. So phi bar monotonically increases with phi. We can write down this relation between them and integrate to obtain the relation between the old and the new coordinate. So then what we want to do is we want to write down a dynamical equation in terms of the new coordinate instead of the old coordinate. Just a few other technical steps. The Blake tensor is going to be written in 2D Fourier space. So if you see Qx and Qy, that's just because I'm writing down the Blake tensor in 2D Fourier space. The Z component is left in real space. This next thing, this separation of timescales, this is a key part of the coarse graining process. So we're thinking about a metachronal wave emerging, and I'm looking at the dynamics of this metachronal wave. I'm not looking at the process of how that metachronal wave emerges. So what I can do is I can average over fast variables. So if I rewrite the equations in terms of mean phases and phase differences, if you take any pair of rotors, they're going, the beads are moving around with some fast time scale compared to the evolution of a phase difference between any two pairs of rotors. So I'm going to average over fast variables, so mean phases, and that reduces the number of variables that we have to deal with. And again, if we're thinking about a metachronal wave, we're thinking about phase differences, and that's what we're interested in. Just a change of notation here. The dynamical equation here is labeled with I to index each rotor. I'm going to start relabeling it with R, little r, the big R's in the Green's function here, they are continuous positions in space. These little r's here are discrete points on the lattice. So we've put the rotors on a surface. These little r's are the lattice positions. So it's not like a sudden change from discrete to continuous. It's a change in labeling. These r's are lattice positions. And when we do that, we can rewrite the dynamical equation in terms of the new coordinate, and then so we've got the intrinsic velocity, and then we've got the effect of the hydrodynamic interactions, which is this big sum and integral. The integral is just because we're expressing the Green's function, the Blake tensor, in Fourier space, so that's why we've got this term here. But the sum is over all lattice positions on our 2D lattice. This we've got it written in what's inside this is this sum is some coefficient that encodes geometric and forcing information, and then we have cos of the phase difference and sine of the phase difference. And at the moment, this is done to leading order in A over L, where A is the trajectory size and L is the spacing. So this is done to leading order in the trajectory size. And if we do this, then what we find is that these coefficients only pick up the second harmonic. So the second harmonic in the forcing. So if you remember, we wrote the force as a Fourier series, and we're saying, okay, which components of force matter? It turns out that only the second harmonic of both the force and the friction are the only harmonics that come into this dynamical equation when we do this time averaging step. And the reason for that is there's a symmetry in the problem when we're only going to leading order in A over L. So if I'm going to leading order and I'm sitting on my rotor, if I'm a cilium here, but I'm looking at my neighbor on this side and on that side, then I can't see any spatial difference between these two because I haven't resolved for the size of the cilium. So that's got some symmetry implications, and I'll show you what happens if we then go to the next order and we can then resolve the size. So to get to the dispersion relation, I say, okay, I want to look for a metachronal wave. A metachronal wave has this form. So we've got omega t minus k dot r. That's our metachronal wave. And let's put a perturbation in to see what happens there. So for a dispersion relation, we just expand to 0th order in the perturbation. And we get this term here. The sum over G is the sum over reciprocal lattice space instead of the sum over real lattice space. But I'll show you some plots because it's easier to understand it looking at plots than looking at some horrible looking sum over reciprocal lattice space. So this is the dispersion relation for a particular set of parameters. This one's got the top chopped off, and you can see the top in the inset. It's not a singularity, it's just a high peak. And what this shows us is at a small wave number, we get a higher frequency of the traveling wave than at larger wave numbers. So a small wave number means that neighbors have, they've got their phases quite close together. So the flow created by one cilium is in a direction that helps the other cilium move faster around its trajectory. So we get a higher frequency. So and then if we've got a larger wave number, the difference between the neighbors is such that the flow created by one doesn't help its neighbor so much to speed up around its trajectory. The colors are related to stability, and I'll get to that in just a sec. So with the wave stability, we expand the dynamical equation to first order in the perturbation. So we say, let's look for a metachronal wave, let's put a perturbation on. Expand to first order, we get this linear stability analysis. So what can we say about this? If this prefactor is positive, then this perturbation is going to grow. So the perturbation, so the metachronal wave is unstable. If we have, also I need to just explain the k's and the q primes because this gets very confusing as well. What I'm looking at here is I'm saying, suppose we have a metachronal wave with wave vector k. So k represents the wave number of our metachronal wave. And now we have a perturbation on it, and the perturbation has wave vector q prime. So k is the wave vector of the metachronal wave, q prime is the wave number, the wave number, the wave vector for the perturbation. So if this thing is positive for any q prime, any perturbation wave vector, then this metachronal wave of wave vector k is going to be unstable. If for all q primes, this thing is negative, this coefficient is negative, then we have a stable metachronal wave. So what we can say is that if so, the blue regions show stable wave numbers, yellow regions show unstable wave numbers. What we can say is that if we're trying to predict what kind of metachronal wave could emerge, it's going to have to be something in one of these blue regions. If the metachronal wave is unstable, we're not going to see that emerging in the long time limit. We can compare with some simulations that are based on the dynamical equation, the initial dynamical equation that we wrote down. So these were done by Fan Long Meng, my collaborator, and basically what we find in these simulations is that the emergent wave that comes out is in one of these blue regions. Now, a big thing you might notice about this is that we get a blue region in the forwards and the backwards direction. And if you look at a particular organism and you look at the metachronal wave, it has a fixed direction, and that might change with viscosity or something like that. But if you do an experiment over and over again, you're going to see a fixed direction, whereas here we're not seeing that fixed direction. And that is to do with that symmetry that I mentioned before with the second harmonic. So if we have a second harmonic, we've got this symmetry. So how are we going to see some symmetry breaking effect? Well, as I mentioned before, if you only go to leading order in the expansions, then you can't resolve between the positions of this neighbor versus that neighbor. So you've got this symmetry involved. But if you go to the next order in A over L, so you start to resolve for the size, so you start to see the size of the trajectories that my neighbors are going around, then if they are slightly different distances from me, that gives a symmetry breaking effect. And we can do all the same steps again. So the same coarse graining steps, which involve the time averaging, but this time we just expand to higher order in the trajectory size. Then what happens is we end up picking up terms that include the first harmonic. So all these M's, S, K's, J's, if it's subscripted with a 2, it means that it's got second harmonics in it. If there's a subscript with a 1, it means there's a first harmonic. So now if we do to next order in the size of the trajectories, so we're going from like a very far field approximation to some near field terms. They're not very near field, but they're slightly nearer field. Then we pick up the first harmonic, and that's important for a symmetry point of view. So again, we can do, and because with the first harmonic, we don't have this symmetry, this forwards backwards symmetry that we do with the second harmonic. So this is just an example here of picking some parameters with just the first harmonic. If we just include a first harmonic in our forcing profile, then we see a symmetry breaking here. So we can see that the stable region, so the region where we expect the metachronal wave to emerge, is only in one direction. Now, this is just another example where we put in both the first harmonic and a second harmonic, but we've cranked up the second harmonic, and only have a very small contribution from the first harmonic. So we see something that's closer to the symmetry of just having the second harmonic. But again, if we change the numbers, so the first harmonic has a stronger presence and the second harmonic is less dominant, again, we see the symmetry breaking effect more strongly, more pronounced. These are just some experiments on a single cilium, looking at the beat of a single cilium and making some estimates of what the force is, to try and say, well, what should the beat pattern look like? And that's something that can be different for different organisms. But just the thing to say about this is that this data suggests that there's the signature of a second harmonic in this data, but actually what that data corresponds to in the model that I've written down is actually the component in just one direction. So when you do the projection, you pick up this extra sine phi term. So actually a second harmonic in this experimental data would correspond to a first harmonic in the model. So that suggests that a first harmonic should be present in this model. So it's good to be able to pick that up. But we also see these higher harmonics as well. And the model doesn't pick up anything other than the first and second harmonics because of some of the approximations made. To do with distances, and we need to make those approximations to ensure that the hydrodynamic approximations are valid. So to conclude, what I want to say is that cilia can coordinate their beat cycles into metachronal waves through hydrodynamic interactions. So that was something that had been seen before in simulations. But what we're doing here is we're using an analytical framework to make the connection between the force profile and the geometric parameters and the emergent metachronal wave. And what we can see, some of the key things we can see through this is that it's the second harmonic that's the dominant term for coordination. So when we just did the leading order expansion, it's the second harmonic that came out. So that's the dominant term for getting this hydrodynamic coordination. But if you don't have a second harmonic, you can still get hydrodynamic coordination through the first harmonic. But what is particularly important about the first harmonic effect is that that is what can break the symmetry between the forwards and backwards directions. So when you're doing this kind of analytical framework, it's really important to include the terms that can break that symmetry if you want to say something about the direction of the metachronal wave. And that's also something that is backed by the experimental results. I would like to thank my collaborators who worked with me on this. So this has been a project, I guess it started a long time ago, and then we picked it up again a few years after starting, and then, you know, we've come and gone, and so it's been good to get to this point now. And yes, thank you to the funders as well. And thank you for your time. I'd be very happy to take questions.